Hello and welcome back to chapter 15. Today we're talking about DNA, so we're going to do a little recap. We're going to talk about genes, and we're going to talk about how we make a copy of our DNA. What does DNA synthesis actually entail? So we're going to start off with what are genes made of. We're going to talk about early kind of hypotheses in DNA replication, and then we're going to talk about what actually goes on. How is DNA replicated? So, everything that we've talked about so far, classical genetics, Mendel's rules, sex linkage, all that fun stuff, right? We discovered all of that without really understanding the molecular nature of genes. We didn't know how genes were copied. We know how they were passed on. We knew the ratios. We knew all that stuff, but we didn't understand how do we actually make a copy of our genetic material. And that's where all of the studies kind of turn to focus on this nature of genetic material. So how are genes copied? And when genes are copied and things go wrong, how is that repaired? How do we fix those mistakes? So a little bit of recap on DNA, just for us to remember. It's been a while since our chapter on nucleic acids. DNA is double-stranded. The individual subunits are called deoxyribonucleotides because they're a deoxygenated ribose sugar, a phosphate group, and a nitrogenous base. So that's your A, T, G, or C. Now, whenever we link together these deoxyribonucleotides, we join that sugar phosphate backbone together with a phosphodiester linkage. We'll have a free hydroxyl, which remember, that's an OH, which indicates the 3' end of our DNA strand. And on the other side, the 5' end, we have a free phosphate group. Now, we talk about DNA with two major features. We have the backbone, which is that sugar and phosphate groups that are all phosphodiester bonded together. And then in the middle, we have our series of bases. And again, that's your A, T, G, and C. Okay, those are your nitrogenous bases. Now, that's if we're talking DNA. We're going to talk RNA in the next chapter, so I'll go ahead and mention it now. Remember, there are no T's in RNA, so instead we see the base uracil replace thymine. So here is our individual deoxyribonucleotide. We have our sugar, we have some sort of nitrogenous base, and we have our phosphate group attached to the other side. Now, when we're polymerizing DNA, we add the sugar and phosphate backbone together, and again, we get a bond between the phosphate group and an OH group. Okay, that's what's going on here. Now, the 5' end has that free unbound phosphate, and the 3' end has this free unbound OH group. And in the middle are the nitrogenous bases. So in this section, we have a T, we have an A, and we have a G. Now, remember when we talked DNA, we mentioned that each strand has polarity, which means the ends are different, right? We have a 5' end with that free phosphate, and we have a 3' end with that free hydroxyl, that OH group. Now, this is going to be very important, okay? This directionality is very important when it comes to DNA synthesis as well as transcription translation. So pay attention to the directionality when we talk about it as we go. As we mentioned before, Watson and Crick, and really Rosalind Franklin, let's not forget her, were the two who proposed this structure of DNA. They were the ones who figured out that these strands, our sugar phosphate backbones, run anti-parallel to each other, which means our directions, they run in opposite directions. So if one strand is running, say, 3' to 5', okay, in one direction, the other strand runs in the opposite direction. It will run 5' to 3'. Okay, that's what we mean by anti-parallel. They are parallel to one another, but they are moving in opposite directions. Now, those antiparallel strands twist together just like a Christmas ribbon to form that double helix. So here's what that looks like on our left side here. This is what it looks like untwisted. So we have those sugar phosphate backbones. You see they're running in opposite directions. And then we have our nitrogenous bases in the middle. And you'll notice they're following our base pairing rules, right? A always binds to T, and G always binds to C. Those are our rules. And you see all those rules are being obeyed. Now on the right, exact same thing, except now we're showing that double helix, that lovely twisting. Just to reinstate our base pairing rules, here they are. And these, once again, are going to be very important as we talk about synthesis and as we talk about transcription and translation later. So if you don't already, please know your complementary base pairing rules. Now, that double helix is stabilized and held together in a few different ways. Not only is it because of that complementary base pairing that holds those two strands together, but we also get interaction between the base pairs inside the helix. So not only are they bonding to each other, but they're interacting with other base pairs inside of that helix. So there were some early hypotheses about DNA synthesis, mostly proposed by Watson and Crick, and they believed that our existing DNA strands served as a template. So essentially, one strand of our DNA, would be the template, and through the base pairing rules, we would be able to use those rules in order to build our new strand off that template. So A binds with what? T. T binds with what? A. And C binds with what? G. Okay, so we used our template in order to build our new strand. And remember, they're running anti-parallel, so our new strand is going to run like this. Okay, so we're using a strand, we're using what we already have to make our new DNA. However, there were three hypotheses for how this takes place: semi-conservative, conservative, and dispersive. So for each of these hypotheses, if we're talking semi-conservative, this is where our parental strands separate, and each of these strands is used as a template to create our daughter strand. What we're going to see with this is that each daughter has one new and one old. Okay. For conservative replication, the parental molecule is pretty much going to just serve as the template and create an entirely new daughter molecule. So at the end of the day, we're going to have a daughter with old strands, so basically the parent strand, and the other is going to have two new strands. Lastly, we have dispersive. This one is the most dispersive of the hypotheses because the parent is going to be cut into tiny, tiny little pieces, and then our daughter strands are going to be a mix of old and new, kind of Frankenstein's monster, if you will. So let's draw out what that looks like. For our conservative, this is our parent strand. We're going to always start with the parent in red, and I'm not going to draw out the direction. But, remember they're anti-parallel. So that's our parent strand. And what this is proposing is when we create our daughter strands, one is basically just going to be the parent strand. We used it as a template. And one is going to be our entirely new daughter strand, that we made. So that's conservative. We're taking our parent template and we're pretty much keeping it the same. We're just using it to make an entirely new double-stranded daughter. Now for semi-conservative, start with our parent strand in red once again. And what's going to happen here is we are actually going to split apart. We're going to split apart that parent strand. We're going to break the hydrogen bonds holding together our base pairs. So we're going to see one parent strand for each, and we are going to use that parent strand as a template to create our new daughter strand, just like so. So you'll see for our two daughter strands, they are each half old, half parent, and half new. So that's why we're calling it semi-conservative. Now, dispersive, okay, again, this is our most kind of destructive form. So we have our parent strand in red, and what's going to happen? We are going to see that parent strand cut apart. So we're going to just break it into pieces. And what's going to happen is our two daughter strands are now going to be a mix. Okay, truly a mix of both. And so you can see our two daughter strands are now truly a Frankenstein's monster. They are a mix of old and new completely. Now, as we continue on, we're going to talk about DNA synthesis and how it happens. But I want you to kind of keep in mind and ask yourself, which hypothesis did we find support for? Was it conservative? Was it semi-conservative? Or was it dispersive? So, the other thing I want you to keep in mind as we go is who are the key players and what do they do? Okay, that's going to be a big thing for this chapter. Okay, what's the enzyme and what is it doing? So we're going to start off with an enzyme called DNA polymerase. And this is going to be a very important enzyme because it is what's actually building our new strand. Okay, it is catalyzing DNA synthesis. And we typically have a few different types of polymerases, and I won't make you learn each type. So just know in general what a polymerase does, a DNA polymerase. But the important thing about them is that they can only work in one direction. Okay, they can only build those phosphodiester bonds. They can only add in those subunits to the 3' end. So that means when we are building a strand of DNA, it is always going to proceed in the 5' to 3' direction. Okay, because new bases can only get added on to that 3' end. Now, DNA polymerization is an endergonic reaction, which means it does require energy, right? We have to put energy in order for this polymerization to occur. And that's where our monomers come into play. So the monomers, the individual subunits that we're adding on to make our new DNA strand, are a little bit different, okay? They're not just a standard deoxyribonucleoside. What they are is a deoxyribonucleoside triphosphate. Okay, triphosphate, 3 phosphate groups. We've seen that before, right? With ATP, cellular energy. Okay, so they're coming back. Okay, we are adding these bases with all of these phosphate groups because they have extremely high potential energy. Remember, anytime we have a bunch of phosphate groups together, they're all negatively charged. They don't want to be there. And so they are going to have very high potential energy. They're going to release a lot of energy. So whenever we're adding in these monomers with those phosphate groups, when we pop off those phosphates, enough energy is released in order for that phosphodiester bond to form. So here's what we're seeing here. Okay, here is our DNTP. So this is just our monomer with those extra phosphates right here. Okay, when we do a synthesis reaction, when we're joining it together, we can pop off those phosphate groups and it releases enough energy in order to form that phosphodiester bond. That's typically endergonic, right? That needs energy to be made. So where does replication start? We're going to draw out our little strands again, just to keep up. And remember, don't forget direction. Direction is important. So we got our anti-parallel strands, one running in one direction, one running in the other. And the first thing that we can do before we can make a copy of our DNA is we have to split apart these strands. Okay, we have to be able to access the bases in the middle because remember, there's bases in here being held together by little hydrogen bonds. So we got to break those hydrogen bonds. And that's what we're going to do. Okay, and we break those hydrogen bonds at an area called the replication bubble. Okay, we're essentially just forming a little bubble. Okay, we're breaking apart those hydrogen bonds, and those bubbles are going to form at something called the origin of replication, okay, the start. And this is identified by just a very specific DNA sequence. This is going to tell all of our enzymes, this is where we start, okay? This is where we need to start pulling apart those hydrogen bonds. Now, when we pull apart, when we break those hydrogen bonds, and we're going to see this more in the picture, it's going to look a little something like this. We have our double strands, and you'll see, okay, they're broken in the middle here. We've kind of pulled them apart. We've made a bubble. Now, this little area right here is called the replication fork. Okay, it's just the region where essentially those bases are still held together, but we're pulling them apart. So as this bubble gets bigger and bigger and bigger, those replication forks are going to proceed bidirectionally. So one's going to go this way and one's going to go that way. They're going to grow in two directions. So here's what that looks like. You can see the replication bubble right here. We can see it down here as well. And this little area right here is the replication fork. Same thing right here, this little area, replication fork. And as we kind of pull that bubble bigger and bigger, that fork is going to move, and we're going to grow this replication bubble in two distinct directions. Now, let's talk about a few more key players. So DNA polymerase, that's what's going to go in and add in the bases, right? That's what's going to read the template and add in the appropriate complementary bases. Now, some more key players, okay, DNA helicase. This is what goes in and actually breaks the hydrogen bond. So this is what's going to go in and separate our DNA strands, okay? Now, once we are separated, okay, our strands don't like to be separated. They want to be joined together. So we have to attach something called single-strand binding proteins, okay, SSBPs. And they are going to attach to those strands that we just separated, and keep them from closing. Okay, it's going to keep them from snapping back together. Now, whenever we're unwinding DNA like this, whenever we're growing that replication bubble, okay, it's going to cause a lot of tension on the regions of DNA, all curled up in that double helix, okay? Lots of tension. So we have an enzyme called topoisomerase that's going to sit right in front of that replication fork, and it's going to relieve that tension. It's going to cut some of our DNA helix and rejoin it in a way that relieves tension and makes sure our DNA doesn't break as we are unwinding it. Now, remember, we have some limitations. DNA polymerase can only work in the 5' to 3' direction, which means one strand of our replication bubble is going to have no problem, okay? It's going to be creating that new strand toward the replication bubble, and as we unwind our DNA, it can proceed no issue. But this is going to give us some trouble with the other existing strand, and we're going to see that. But first, we're going to talk about the leading strand. Okay, this is, in short, our unproblematic strand. Okay, everything works out perfectly for the leading strand. Now, for this, before we can start building our new strand, we have to add what is called a primer. Okay, a primer, just like when you prime a wall before you paint it, right, is something we have to do first. We have to attach a primer in order for DNA polymerase can bind. So usually our primer is an enzyme called primase. Okay, that's what's going to bind to our specific region where we want to start DNA synthesis. So it's going to bind to that DNA template, and it's going to allow DNA polymerase to come in and start building our new strand. So once our primer binds, then polymerase can bind and start adding those monomers, those DNTPs, to the 3' end. Okay, once it does that, it's going to start synthesizing our new strand, and it's going to do it toward that replication fork, okay, toward that little area of our bubble, okay, where things are getting unwound. So it's going to happen just like that. This is called the leading or continuous strand. And the reason why we call it that is because as this unwinds, okay, that strand can just keep getting made on and on and on continuously. So here is that process illustrated. And I think we're going to watch a video too, because it can be kind of hard to picture this. So you'll notice here's our template strand in gray, and it's running 3' to 5', just like that. Okay, so here's topoisomerase. It's running right in front of that replication fork. It is relieving tension. Here's helicase. It's unzipping our DNA, right? It's sitting right at that replication fork and opening up our double helix. And here's our single-stranded binding proteins holding open that replication bubble. Now we have primase, our primer. It's going to bind to a specific region, make a little primer. And then DNA polymerase can bind and start adding in our new bases. So you'll notice, okay, 3' to 5', remember, for our template. But since the new strand we're building has to be anti-parallel, okay, look, it's getting made 5' to 3'. And that's what we want, right? Because that is how DNA polymerase can work. It can only synthesize in the 5' to 3' direction. But this is good. This is our leading strand. So you'll see as helicase unwinds, as it progresses, okay, polymerase is just going to get to follow right along and make a nice, long, continuous strand. Unproblematic, no issues whatsoever. Now, what about our problematic strand? Okay, that is what we call the lagging strand or the discontinuous strand. And the reason why is because of that directionality, right? DNA synthesis has to proceed in a 5' to 3' direction. And so because of the direction of our template, we have to make this new strand away from the replication fork. So it will not be able to proceed continuously all in one long go. Okay, and that's where this discontinuous replication hypothesis starts. So primase is going to bind right at that replication fork. Polymerase is going to bind right behind it, and it's going to start making a new strand, but that's going to happen away from that replication fork. So as that replication fork keeps going, we're going to end up with a gap. Okay, and so with this, we end up making our new strand in a bunch of little fragments instead of one nice long new strand. So here is what that is showing. Here is our lagging strand right here. You can see our leading strand easily being made toward that replication fork as our DNA unwinds. But you see with the lagging strand, we have to go in the opposite direction because of those directionality rules. We always have to proceed in the 5' to 3' direction for our new strand. So primer binds, okay, DNA polymerase would start adding in new bases toward 3'. But as our DNA unwinds, look what happened. We got a gap, right? Because our leading strand, right, unproblematic, can keep working as our DNA is unwound, no problems. But because of this directionality issue, the lagging strand, okay, is going to have a large gap here. And these little fragments that we're creating, these small little pieces, are called Okazaki fragments. Okay, short strands, and they are unique to the lagging strand. Okay, lagging strand is made in these short, discontinuous Okazaki fragments that will later be joined together after they are finished being formed. So here is what that looks like, just focusing in on our lagging strand. So we have all the kind of key players. We blocked out the leading strand, but topoisomerase is still here, helicase is still unwinding, still got some single-stranded binding proteins, but you'll see our primer binds. Okay, polymerase starts making our new strand in the opposite direction of that replication fork. And so that's our first fragment. Then it's going to happen again. Primer's going to bind. Second Okazaki fragment's going to get made. Then we are going to get a new player, okay, DNA ligase. Ligase, it's going to come in, and it is what's going to join together those fragments. Okay, ligase is like a glue stick. It's going to come in and join together those small fragments. So you'll see this lagging strand has to be made in a series of fragments that will be joined together. And you'll notice that's what we're saying here. We have ligase that's going to catalyze a phosphodiester bond. It's going to come in and it's going to join the backbones of our two Okazaki fragments. So here is a great little summary table of all of the different players in our protein synthesis game and what they do. So make sure you're able to identify them not only visually, but based on kind of their description. Okay, what do they do? So here it is all in one go, okay, because this is all happening at the same time. Leading strand is being made, the lagging strand and the Okazaki fragments are being made, our helix is getting unzipped, all of this is going on all at once. And we call this whole entire thing the replosome. Okay, it is a DNA synthesizing machine. Okay, I just wanted to show you guys a short bit of this video. So we have our template strand here. These are the individual subunits that we're going to get added on. Here is our primer that binds to our template strand, so RNA primer. And then we have DNA polymerase that's going to come in and it's going to start adding on those dNTPs, those subunits, to our template strand. So this is the synthesis that we're talking about. This is RNA polymerase going in and adding in the appropriate bases to create our new strand. Now, let's see what this all looks like together. So it's going to proceed 5 to 3. Here's our DNA double helix. Okay, the first thing that we want to do is we want to break those hydrogen bonds. So here's the replication bubble. Okay, you can see we've broken the hydrogen bonds. We're looking at the single template strands. We have the replication fork on both sides, and you'll see those strands, once again, anti-parallel. So we have helicase that sits right in that fork, and what's it going to do is it's going to unwind our DNA. Okay, it's going to proceed in each direction, and it's going to grow that replication bubble in both directions. These are the single-stranded binding proteins. So these are the little things that are going to bind and hold open that replication bubble that we just formed. And you'll notice, okay, we're getting some tension on either side once we unwind that helix. And so topoisomerase is going to come in, it's going to cut, and it's going to rejoin in order to fix that. So we have our primase coming in, adding in our primer to each of our template strands here. Okay, then we're going to have, okay, zoom in here, we're going to have RNA polymerase bind, and you'll see as this molecule, as this replication bubble gets bigger, okay, our leading strand here is going to grow continuously toward that replication fork. Super unproblematic. But you'll notice with the lagging strand, since it has to go in the opposite direction, we're going to get this short set of fragments. Okay, so our leading strand, nice and long and continuous, and our lagging strand in short fragments where we keep adding these primers on and on and on over again to create our Okazaki fragments. Now, once we are ready, okay, the Okazaki fragments will get joined together, okay, using ligase, which is like our little glue stick. Okay, it'll come in one by one and join together those fragments by joining together their phosphodiester bonds, so DNA ligase. So as we said before, this replosome is pretty much that entire thing, that entire unit that we just described. Okay, all of the enzymes for DNA synthesis, all of that replication bubble, the replication fork, and we describe it as one kind of macromolecular machine, okay, that's just doing DNA synthesis. Now, we have learned recently that some things we previously knew about the replosome may not be correct, okay, or may be different. So we believe that the replosome is a lot more dynamic than we thought it was, so it might be changing based on the needs of DNA synthesis, and the continuous strand might not be as continuous as we once believed, which is new, and that rates of DNA synthesis can actually vary widely. So it's not always going to be occurring. It's going to be occurring when that cell needs to replicate its DNA and when it needs to divide. Now, one thing that we always have to mention when we're talking DNA replication is something called telomeres. Okay, telomeres are the region at the end of our chromosomes, so at the end of our DNA sections, and they're very important because they are essentially junk DNA. Okay, and the reason why we have to have some sort of junk at the end of our chromosomes is because the leading strand, once again, unproblematic. Okay, it can be made all the way till the end, and you're going to get a full double-stranded copy, no problem. However, our problematic strand, our lagging strand, okay, because of the way that it's made, the final fragment isn't going to make it till the very end of our DNA. Okay, DNA polymerase cannot add to the end without a primer. So what you end up getting is a little bit of DNA left at the lagging strand, and that DNA is usually eventually degraded. So it goes away, it disappears, it vanishes. Okay, so when that DNA vanishes, and it's usually about 50 to 100 nucleotides, okay, we want the DNA that vanishes to be junk, right? We do not want our chromosomes to disappear, the actual important information. And over time, our linear chromosomes would vanish because every single time that they are synthesized, that they are replicated, we lose a little bit of them. Okay, so telomeres are kind of like the junk at the end of our chromosomes. Okay, so when we replicate, we will just lose some junk. Okay, short repeating stretches, instead of losing the actual important information in our genes. So here is showing that process. You'll see leading strand makes it to the end, no problem. But with our lagging strand, just because of this function of the little fragments, we get this little overhang, right? A little overhang of unreplicated material. Too short to add another primer to. So this little stretch would be lost eventually. It would be shortened. And again, we don't want that loss to be of our actual important genetic material. Now, what happens with telomeres, right? They degrade over time. And it's actually believed that telomeres are related to aging because telomeres are not endless, right? Once we get rid of the telomeres, once we've divided enough, once we have essentially done enough DNA synthesis, okay, our telomeres would be gone. And then with every subsequent cell division, we're going to start eating away into that chromosome. Okay, so that's a problem, right? And that's actually associated with cellular aging. So up until a point, you're going to hit where your telomeres are gone. And then once we start eating into the chromosomes themselves, you're going to see the effects of cellular aging. Now, if we can figure out how to repair our telomeres, right, would that give us immortality? Right? Would it make it so that our chromosomes were never degraded and we would never see signs of aging? Okay, is there the secret to immortality? And we actually know that there is a way we can repair our telomeres. Okay, it is an enzyme called telomerase. Now, what it does is it's going to bind to that overhang, and it's essentially going to kind of make that overhang just long enough so that we can continue with DNA synthesis. Okay, so it's essentially going to kind of repair our telomeres. So here's telomerase coming in. It's adding a few bases to that overhang so that polymerase can do its job like it should, and we actually don't lose any of our important chromosome or any of our telomeres. So interesting, but telomerase is not active in most body cells. In fact, telomerase is only found in gametes and stem cells. Okay, all of the cells in your body do not have telomerase. So as you age, your chromosomes are going to get shorter and shorter and shorter. Now, this led to the idea that you are limited by a certain number of cell divisions. Okay, there's a certain number of cell divisions that all of your cells can go through before your telomeres are gone and your chromosomes are going to start shutting down. Okay, they're going to get shorter and shorter, and suddenly the recipe is gone, right? You cannot make any of the things you need to make because your genetic recipe is gone. So, how can we figure out how to turn on telomerase in our cells, right? That would be the key to immortality. And one of the most kind of tragic and poetic things is that most cancer cells, even somatic cancer cells, have active telomerase. Okay, somehow cancer cells have figured out how to turn on telomerase and repair their telomeres. Okay, repair so their chromosomes will never shorten. Because remember, the whole thing about cancer cells is unregulated cell growth. So if their chromosomes were to shorten, the cell would no longer be able to replicate and divide. But somehow they are turning on this telomerase, right? Can we figure out a way to inhibit telomerase to stop cancer? Or can we figure out how to turn on telomerase in our healthy cells in order to combat aging? And the poetic thing, I think, is that the answer to immortality may lie in the very thing that is killing us. Very dramatic. So the last thing that we want to talk about with DNA synthesis is mistakes, right? What happens if something goes wrong? And we're going to start off talking about DNA polymerase, because DNA polymerase, right, the enzyme that's going in and adding in the bases, not only is it fast, but it's pretty accurate, okay? And it needs to be accurate, because if we make a mistake, okay, these mistakes can be disastrous, right? Any change to our DNA sequence is called a mutation, and a lot of mutations result in things like the development of cancer. Now, not all mutations are bad. We're going to talk about that, but a lot of them are. And so accurate replication is really life or death. It is very important. And thankfully, DNA replication is very accurate. Okay, DNA polymerase is going to match bases. It's going to follow those base pairing rules with very high accuracy. It really only will insert an incorrect base about once every 100,000 bases. Okay, because it's just better to have a correct base matching. Okay, it's the most energetically favorable pairing, and if bases are not matched correctly, okay, that's going to cause a very distinct shape. Okay, correct bases have a distinct shape, and if you make a mistake, you're going to see a very distinct shape as well. So, there are a variety of systems. Okay, so say some mistake does happen, we have a lot of enzymes and different systems in place that can remove those mistakes and replace them with correct bases. Now, one of those is DNA polymerase itself. I know it can do it all. Not only does it add in new bases, but it can also proofread its work. Right? If it makes a mistake, it can recognize that, it will remove the mistake, and it will add back in the correct nucleotide, okay, the correct base. So it can do this because it has something called an exonuclease active site. And we're going to see it in the picture, but it's essentially just a site within polymerase that it can remove and shove that mistake, that nucleotide that's not supposed to be there, into this exonuclease active site, and then it'll add back in the correct base. So here's what that looks like. Here's our polymerase. Here's our big green box. You'll see it made a mistake, right? It added an A to a C. That's going to cause a little bulge in our new strand. Okay, it's an incorrect base pairing, very distinct shape. So what it can do, it can shift that wrong base into that exonuclease site. So it can essentially just kind of shove it over, and then it will add in the new base. Okay, it will replace it with the correct base, which is a T in this case. Now, if DNA polymerase failed, okay, if it didn't recognize its mistake, if it didn't proofread correctly, we still have other systems that can correct things. Okay, one of those is called mismatch repair. So this is after DNA synthesis is complete, after polymerase has done its job, okay, incorrectly, but it's done its job. Now this mismatch repair can go in, it can recognize a mismatch pair, it can remove that section that's incorrect and fill it with the correct bases. Now, not only do we see mistakes and mutations because of errors in DNA synthesis, but our genes can actually be damaged by external things. So genes are kind of under constant assault. And one of the best examples is sunlight. Okay, DNA can actually be damaged by high energy light, like sunlight, X-rays, chemicals, all sorts of stuff. And there, once again, are a series of systems that can go in and can repair DNA damage from things like UV light. Now, we're going to use UV light as our example. Okay, so this is your PSA not to go to a tanning bed. Okay, UV light is very bad for you. And what it can do is cause something called thymine dimers. Okay, these dimers, essentially two thymine bases, will join together, and it makes it so that there's a little kink, a bump in our DNA strand. And when that happens, okay, it will block DNA replication, which is not a good thing. So if you've ever gotten a tan, right, it might look nice, but what actually is a tan? Okay, it's DNA damage. Okay, you have damaged your DNA, and in response, your body is producing more melanin pigment in order to protect itself. So anytime you get a tan, that is a direct result of DNA damage. So always wear your sunscreen, don't go tanning, and you will reduce your chances of DNA damage. So here is our UV light, and you'll see what happened is a thymine dimer formed. So we've kind of found this bonding between our thymines, and we get a kink in the backbone of our DNA, which is going to make it so that it cannot be replicated. Now, what's going to go in and help? Okay, that's our nucleotide excision repair. So they can recognize that kink, which we see from our thymine dimer. They're going to go in, they're going to remove the damaged DNA, and they're going to insert a new healthy strand. And then, of course, our glue stick, DNA ligase, is going to go in and it's going to glue it all together. So here is our thymine dimer. Okay, you'll notice that nice little bent backbone, and nucleotide excision going to come in, it's going to cut and remove the damage, and then, okay, replacement. So we're going to go in, we're going to replace it with a nice healthy strand, no damage, and ligase is going to seal up where we made those cuts. Now, what happens if you have a defect in a repair system? So what happens if your repair system isn't functioning correctly? And there are genetic conditions that cause exactly that. So for example, xeroderma pigmentosum, XP. This is an autosomal recessive, so we should know what that is by now. Autosomal recessive disease that essentially is a damage to this nucleotide excision repair system, okay? It won't work. And what this causes is extreme sensitivity to UV light, right? If now we're getting DNA damage and thymine dimers, but we don't have the systems in place to fix it, you are now much more likely to get skin cancer than someone with a functioning repair system. And this condition is caused by mutations in the nucleotide repair systems. Their cells cannot repair that damage, and again, you're going to see a high rate of skin cancer. Now, any defects in these repair systems are almost always going to be associated with cancer, right? If we have damaged DNA and it cannot be repaired, we're going to see unregulated cell growth, okay? Mutations in genes involved in that cell cycle that aren't fixed, we're going to see tumor formation, we're going to see all of that nasty stuff. So if our mutation rate in a cell is elevated because of these repair genes, very likely cancer is going to be a result.